Helicobacter pylori is a gram-negative helicobacterium that is commonly encountered in the epithelial lining of the stomach. H. pylori is the most common cause of peptic ulcers. It is responsible for over 90% of duodenal ulcers and 80% of gastric ulcers. Over 60% of the world's population is infected by H. pylori. However, in most individuals, the infection is asymptomatic. Those who are infected by H. pylori have a 2 to 6 times increased risk of developing gastric cancer and malt lymphoma when compared to uninfected persons. It's a highly successful pathogen, as it has been able to coexist with humans and adjust to our bodies for over 50,000 years. It's been around for a very long time. This and the fact that it is so common makes H. pylori and studying it highly clinically relevant. Because it's so common, understanding H. pylori is crucial for careers in medicine, pharmacology, and microbiology. Studying it has improved our understanding of chronic infection and the microbiome's role in human health. We also have been able to develop an antibiotic treatment for H. pylori infections, increasing the quality of life for many patients. The structure and morphology of H. pylori is highly essential to its function. There are numerous distinctive bacterial traits that enable its survival in the harsh environment of the human stomach. These features are specific to H. pylori. Features such as its spiral shape, production of urease, motility due to flagellum, and the presence of outer membrane proteins all contribute to this special capability that is surviving in our gastric systems. Its helical shape and 4 to 6 unipolar flagella make it highly motile and allow it to burrow through the viscous mucus layer on the stomach's epithelial lining. Its gram-negative cell wall has a thin layer of peptidoglycan and an outer layer of lipopolysaccharides. This lipopolysaccharide layer helps H. pylori avoid detection by the body's immune system. This allows it to persist in the body for many years. H. pylori also employs multiple outer membrane proteins, such as BABA and SABA, which allow H. pylori to bind to the epithelial wall cells. These proteins are embedded in the bacterial cell's outer membrane and allow for easy adhesion to the viscous epithelial cells. The production of urease allows for the neutralization of the stomach acid. The urease breaks down urea, which is found in the stomach normally, breaks it down into ammonia and CO2. The ammonia then reacts with the gastric acid and raises the pH around the cell, protecting the cell from the harsh gastric environment. Apart from these unique and specialized traits, H. pylori shares many of its remaining features with standard bacterial prokaryotes, such as the free-floating genetic material inside the cell, a plasma membrane, or ribosomes.
H. pylori is a gram-negative bacterium best known for its role in causing stomach ulcers and gastric cancer. In terms of replication, like most prokaryotes, it reproduces through binary fission, a process where the DNA is copied, the cell elongates, and then divides into 2 identical daughter cells. What makes H. pylori unique is that it also uses flagella to burrow into the protective mucus lining of the stomach, giving it access to a more stable environment for growth and division. H. pylori has a very specific growth requirements. It's microaerophilic, meaning it needs a low oxygen environment, about 5% oxygen, and increased carbon dioxide to survive. It thrives at 37 degrees Celsius, which matches the human body temperature, and prefers a slightly acidic to neutral pH, which it creates using the enzyme urease. Urease breaks down urea into ammonia, which neutralizes stomach acid, allowing the bacteria to survive in such a hostile environment. It also requires rich, complex nutrients, including amino acids, vitamins, and trace minerals. Because of the demanding conditions, H. pylori grows slowly, often taking 3 to 7 days to form visible colonies.
There are several microbial control methods such as physical agents, chemical agents, and mechanical removal methods. Physical agents include heat and radiation, which help destroy microbial cells. Chemical agents include disinfectants or antiseptics, which prevent further microbial growth. Then there's mechanical removal methods, which separate microbes from surfaces and solutions like washing and sanitizing. Helicobacter pylori causes stomach ulcers, gastritis, and gastric cancer. This is usually treated through antibiotics like cimetidine or ranitidine, which also helps slow down the secretion of acid that's in the stomach. Therapy can also be recommended where you would take clarithromycin and amoxicillin for 7 to 14 days to prevent any more bacterial infections from spreading, and any stomach acid inhibitors. Sanitation and sterilization is also highly recommended, especially in hospital settings, to avoid the physical spread of bacteria from person to person. Detecting H. pylori can be done through lab tests and with the patient. The urea breath test measures the amount of carbon dioxide in the patient's breath after drinking a solution that contains urea to detect any of the bacteria. The higher the amount of carbon dioxide, the more likely H. pylori is present. The stool test is done with fecal matter to detect if there are any harmful antigens, which are produced by the H. pylori bacteria. The blood test takes a sample of the patient's blood, which also looks for harmful antigens. However, the blood test is more difficult because it can't distinguish between past infections that have cleared from the active infections, since antibodies can still linger in your blood after the infection has left. An upper endoscopy is a more invasive test than the previous ones mentioned. This method uses a scope to withdraw tissue from your stomach as a sample to test for H. pylori to take to the lab for urease testing, which looks for the urease enzyme that is produced by the bacteria. Lastly, the gram stain procedure is performed in the lab to look for H. pylori, which is a gram-negative and has a curved bacillus. These are some of the many tests that can be performed to detect the presence of H. pylori, and there could potentially be even more efficient ways to detect this bacteria in the future with more research and technology.
H. pylori interacts with the human body in complex ways, often evading full immune clearance by staying in close contact with epithelial cells and altering surface antigens. The innate immune system responds first. Gastric cells and macrophages detect bacterial components like flagellin using toll-like receptors, leading to cytokine release, which recruits neutrophils and promotes inflammation. As the infection persists, the adaptive immune system activates. Dendritic cells present H. pylori antigens to helper T cells, which then stimulate B cells to produce IgA and IgG. However, the bacterium resists antibody action using strategies like antigenic variation and immune suppression. This long-term immune engagement often leads to chronic gastritis, increasing the risk of ulcers and gastric cancer, especially in genetically predisposed individuals. Despite a strong immune response, H. pylori typically persists, making it one of the most successful chronic human pathogens.